Good evening, everyone. I'm glad we didn't have a snowstorm today. It was a historic moment when McGill canceled classes, and it just happened to be on the same day that this was originally scheduled. I am Dean of Students, but in my other secret life, I'm a professor of entomology. And it's entomologists, when we see each other on the streets, there's kind of a connection. Because it takes a certain something to spend a lot of time looking at small critters with such a high degree of interest and enthusiasm. And I find entomologists, if you ever go hiking with an entomologist, you only make it about a kilometer, right? It's like, oh, my wife drives her crazy, already looking down at her feet instead of looking at other things out there. So I'm always delighted to think about entomology, and the history of entomology is so rich and fascinating, and I'm really, really pleased that I get to introduce Ehab today, Professor Ehab Abouheif. I have a whole list of things here. I'm not going to read it all, but I'm just going to tell you it's all very impressive. Very, very impressive accomplishments. PhD from Duke University, started at McGill in 2004, is that correct? Research program is really the leading edge of Eco-Evo-Devo, which integrates the fields of ecology, evolutionary, and developmental biology. It's a really fascinating field, and we're lucky at McGill to have some lead researchers in this area. Ehab Abouheif uses the complex societies of ants to uncover how gene-environment interactions influence development and evolution. He has a very impressive CV with lots and lots of citations and great numbers behind them, which is wonderful. But what it actually means is that the work is influential. The work impacts people from around the world and at different stages of education as well. It's very foundational and very, very groundbreaking. One of my most favorite things, a little anecdote about our speaker, is the provost was on a trip with 3 deans, and I think you were outside of Abu Dhabi, and there was a flat tire on the car, and there was a photo posted on Twitter of the provost, the dean of engineering, I think it was the dean of faculty of agriculture and environmental science, and another dean, and Ehab down on the ground looking at ants and talking about ants with senior administrators at McGill, and you could see in their eyes that they understood why entomology is such a fascinating and important discipline. So I'm really delighted to have a colleague and friend here to talk to you about the question that stumped Darwin, ants and evolution, and we look forward to your talk.
It's so wonderful to be here in this the Rare Books Library. This is indeed a rare occasion. There are not many occasions where I have elementary school friends, people, my neighbors in my own condo just from down the street, my own students in my lab, and even my colleagues from my department. So if I blow this, it's like across the board. But really, it's quite an honor to be able to have this time to talk. I really want to give you an idea. What I'm hoping to do is go through some of the last, we've been at McGill, as Chris knows, since 2004, the last 15 years. And I just want to tell you about 3 groundbreaking discoveries that try to answer the question that stumped Darwin, and make the point that really, these discoveries are incomplete without the rich history that we found in the Blackwood Library, and some of the things that we unearthed that we only unearthed actually after we made the discoveries and realized, this is like, people had been talking about this as a really rich period, and I'll go into some of the more details as the talk goes on, but just to really make that you'll see as we go on that there's this conversation between the past and the present, and it makes you wonder, looking forward for the future of biology, whether the new ideas are in the past or in the future. So we should revisit that at the end of my talk. Okay, so the question that stumped Darwin, this is me in the Rare Books just a couple of rooms down. This was taken holding the first edition of The Origin of Species. For an evolutionary biologist, this is like, holding something like incredibly special, and I felt felt as the energy as I was holding it, like almost a white beard. Darwin's white beard would start growing on my face as I was holding it. But here, of course, these are the ants we work on from the genus Phylogenetic. But I want to just step back for a second and just introduce Darwin and the science of evolution very quickly. This is, I asked, Loren to take a scan for me of the origin of species. This is again the first edition that I was holding. And Darwin, there are two major insights from the origin of species. One is that after, basically, and the point that is less often talked about is that all of life is connected. That was really something that Darwin brought together. Before the origin of species, people thought of species on Earth each being specially created, each being independently created. And it was Darwin with his idea, this is kind of his notes, these are historical notes, the notion that everything is connected in the tree of life. And there's only a single, out of the 400 or 500 pages in the origin of species, there's a single figure. And that figure is what's called a phylogenetic tree or a historical tree. And again, this is just an elaboration of these original ideas that were in his notes, basically showing that these are common ancestors and everything, just as we are related to our family, our cousins, our uncles, that all of life is related in these branching trees. And that was really, actually, the most fundamental idea when the origin of species was first published. The second major impact of the origin of species was that Darwin had proposed a mechanism for how this all unrolls. Hey, Bob. And so, this is his mechanism of natural selection. And it was basically the mechanism, the idea that the mechanism would work that would connect all of life together. And so, really, what Darwin left us with is this grand tree of life, connecting all of life as you see it to here. These are all the animals, representatives of all the animals you can see today. Here's the ant, I look forward very carefully. And so this is really our job as evolutionary biologists, as entomologists, and people in the biology department, is to understand how this unravels, how this works. Okay? And so I wanted to do a little exercise, since many of you may not be biologists, just to test out our knowledge of life since we're trying to explain it. And I'm just going to ask a very simple question. How many described species on Earth? So put up your hands if you say A. Okay, don't be shy. I do this in my biology 215 class and they all get it wrong anyway, so it doesn't matter. B, 1 million. C, 2 million. D, 10 million. E, 20 million. So the number of described species on Earth are 2 million. Okay? So this is the number of known and described species. Now it's very interesting because the estimated number of unknown species goes anywhere from 8 to 64 million. So we actually know very little about life's history and diversity. Okay? We've only described 2 million of that, and there's a whole frontier out there that we know very little about. Okay? We need to know it before we destroy it all. Okay, another quick quiz. Which of the following group of organisms have the greatest number of species? A, bacteria. B, protists. C, fungi. D, plants. E, animals. And the answer is animals. Okay? Now, this is the greatest number of species. Now, in terms of biomass, of course, the plants, are much more, in terms of biomass, occupy much more of the Earth, but in terms of the number of species, it's the animals. Okay, last one. Which of the following groups of animals have the greatest number of species? A, mammals. B, insects. C, flatworms. D, clams and snails. The answer is the insects. Okay? So the insects rule the Earth, essentially. And so, if we really want to understand diversity on Earth, we have to understand how, primarily how insects have evolved as well as everything else. Okay, so that's our job. And just to make, moving forward, this was the science of Darwin. he left us with a mechanism and the idea that everything is connected, and we had to figure out the details. Now, something very interesting that connects Darwin to McGill, and this is again through the connections that many of you might not know, but the founding principle of McGill was a renowned geologist, Sir William Dawson. Of course, many of you know Dawson College. That's named after Sir William Dawson. The Redpath Museum has a plaque because the Redpath Museum was founded by Sir William Dawson. Now, the interesting thing that Dawson and Darwin had actually corresponded about geology, and Dawson was one of the founders of the sciences of paleobotany. So he was one of the first people to describe fossils of plants, and sort of describe them. And he wrote 3 books. This is the interesting part that many of you may not know: The Origin of the World According to Revelation and Science, and 2 other books, each of which attacked Darwin. He refuted evolution down to his very core, Sir William Dawson. So the founding principle of McGill was an anti-evolutionist, and he built a museum with the idea that evolution is not a possibility. But now, McGill is of course world-renowned for evolutionary biology, and so the history it's a very interesting kind of history moving forward. So let me just tell you quickly about how we try and answer some of Darwin's questions, how we try and unravel the science of Darwinism, and then we'll get into this interplay of the science. So as Chris mentioned, what we try and do in my lab, and sort of the field in general, is we try and integrate 3 different fields together. And I feel that's the way forward for major discoveries in science, is you have to be integrating things that hadn't been integrated before. And so what we're trying to integrate is 3 different fields: ecology, evolution, and development, in this fancy acronym called ECO, EVO, DEVO. And as Chris mentioned, what really we're trying to do is, you might have heard about many statements in the media, that genes will determine how long we live, genes will determine essentially our behavior, and genes will determine certain types of cancers. We found this mutation that will basically set your destiny to figure out this is how long you're going to live. And the idea of ECO, EVO, DEVO is to take a broader approach and understand really that genes are just one component of the equation, and that genes are real players that interact with their environment. Okay, and so we know, of course, many of you would realize that genes and of course the environment has an effect on everything from the way we develop to the way we behave. But really, it's about the conversation, and this is kind of you'll see it's a very complex interaction between genes and the environment, and that's what ECO, EVO, DEVO is trying to understand, how this interaction affects the way we develop and the way we look and the end. Okay, so what are the main tenets of ECO, EVO, DEVO, just to go through very quickly. And the basis of the field really goes back, the modern basis of the field, as you'll see, it actually goes back a lot further, goes back sort of a revival of ECO, EVO, DEVO, goes back to the discovery of genes that regulate development, and that was foundational discoveries by American E.B. Lewis and 2 Germans, Christiane Volhard and Erich Wieschaus. And they worked on this, the humble fruit fly. And with this fruit fly, they were able to understand the actual genes that regulate development. And so here's an example. Here's a fruit fly embryo. Here's the head of the fruit fly, and there's the tail. And these are, what you see in green, are the expression of this gene called engrailed. And engrailed marks where the future segments of the adult fruit fly will be. Okay? And so you can actually visualize and you can see where in the embryo these genes are going to be expressed and where this will happen. The important thing is, after the discovery of this, many American and Swiss scientists discovered that these genes in the fruit fly are actually conserved in all animals, including humans. So every single living thing on Earth, every multicellular thing on Earth has these genes. And so if you look at this gene engrailed, again, this is the fruit fly, but the fruit fly embryo is filleted. This is the head, this is the tail, and again, it's marking the future segments. Whether you're talking about a fruit fly, a beetle, a grasshopper, a crustacean like a crayfish, or a vertebrate like a zebrafish, and in mice, you see this gene is present in their genome and it's expressed in a conserved way. And then just to kind of talk about the Hollywood experiment by the Swiss biologist Walter Gehring. Here when you show these different eyes to a evolutionary biologist, this is the eyes of a fly. We call this a compound eye. This is essentially made, each of these is a receptor, and each of these are kind of all together, these hundreds of what are called ommatidia send visual signals to the brain and you form the image. This is, of course, the camera eye of a vertebrate, including those of humans. And in the cephalopods, they also evolved a camera eye that's built slightly differently. But these, evolutionary biologists thought that, of course, these are completely separate inventions in the history of life. They were invented on completely different occasions, on different branches in the tree of life. What Walter Gehring found is that even though these are very kind of different constructions, they're regulated by the same gene called PAX6. So the same gene regulates these eyes in fruit flies, humans, and in cephalopods. Okay? And then he did the remarkable thing where he took both the human and mouse PAX6 gene and he inserted it into a fruit fly and he expressed the mouse gene on a fruit fly wing, and he got an eye to grow on the wing of a fruit fly. Okay? And so that shows the actual remarkable conservation of these genes, the function and expression, that you could take something from a vertebrate, put it into a fruit fly, and you can actually get it to develop its eyes. So that's really the remarkable thing. So basically the idea from a typical evo-devo perspective is that it's the changes in the sequence, expression, function, and numbers that lead to all, the form and function that we see today. Because it's a real paradox. I mean, if you have the same genes in all these animals, expressed in the same way and function in the same way, well, how do you make a fruit fly? How do you make a human? How do you make a frog? How do you make something with 5 fingers, with 8 fingers, and so on? And so the idea that we work on, the theories, is that, of course, it's changes in the sequence, expression, function, and the numbers of these genes that give rise to all this form and function. The one thing that's been left out is the eco part of it, the idea that the environment has an influence. And this has been known for a long time, but hasn't really, kind of been integrated, and that's where eco-evo-devo comes in. And this is just to show here on this x-axis is temperature, and here's the number of facets. This is on the fruit fly eye. And you can see here, just if you change the temperature in which the embryo is grown in, you'll get a dramatically different number of facets. And so just changing some aspects of the environment can lead to dramatic changes in the way things work. Okay? And that basically now brings us to eco-evo-devo. And so the way we study eco-evo-devo is, of course, through the ants. And this now is what keeps us busy night and day, my lab. And so it's pretty remarkable diversity. All ants, there's 15,000 species of ants. They all belong to a single family, the Formicidae, and they're remarkable. Just belonging to a single family, they are really diverse, as you can see here in this picture. And you can see a remarkable array of different jaw forms, head shapes, eye shapes, all kinds of crazy things. And you could just, they have a slideshow on the internet on antweb.org, and you could just watch it all day. It's like a slide, the slide, just flips back and forth, as Chris says. So anyway, just to say, E.O. Wilson, you have to take this with a grain of salt, but Bert Holdobler and E.O. Wilson, 2 very famous Harvard biologists, made the comparison. He said if you took all the ants in the world and made them into a single blob, and all the humans in the world and made them into a single blob, that they would be equivalent blobs. Okay? Just to show you how remarkably successful the ants are. And so we've chased these ants all around the world. These are the wood ants in Germany. We go to Arizona. We dig pits in Germany. We risk our lives digging in random fields in Texas. We're afraid to get shot. In the fields of Long Island, and in the tropics here, we're digging up a leafcutter ant nest in Panama. Okay, and of course the reason why the ants are so successful is because, first of all, they're what are called eusocial. So they form incredibly remarkable forms of cooperation. Okay? And so there's often a queen who does all the reproduction, and the workers that are often have a very reduced fertility that help do all the work. And they often cooperate in very complex tasks. Another reason why they're so successful is because of something called polyphenism. So ants are polyphenic. It means when a queen lays an egg, it's kind of like honeybees. You know, when the honeybee, as an egg eats the royal jelly, it'll become a queen, and if it doesn't, it becomes a worker. So the nutrition can very decide which developmental path it will go, and it's the same thing with the ants. Just depending on the nutrition, the temperature, or the social interactions, it will become a queen or a worker. Okay, so now the question that stumped Darwin. We're finally here, just to give you an appreciation. And so you would think, well, why would we even look to the past? I mean, there's no point. I mean, with all these amazing discoveries of genes that you can transform eyes on wings, I mean, why would we even look even past 5 years? You know, I mean, it doesn't make any sense. But yet, this one question by Darwin has been unanswered since 1859. And this is, again, thank you, Lauren, for this scan. But if you just read this paragraph right here, Darwin really, this paragraph challenged Darwin's whole entire theory of natural selection. Okay? It was the one of the things, he had a number of issues that challenged him, but this was the most, what he called the climax of the difficulty. And he says over here, but we have not as yet touched on the climax of the difficulty, namely the fact that the neuters of several ants differ, not only from the fertile males and females, but from each other sometimes almost to an incredible degree, and thus are divided into 2 or even 3 castes. What is he talking about? What he's talking about is this. Because, you know, if you look at, here's a typical sort of ant colony that we're used to looking at. It has a typical queen that's quite a bit larger, and all these, you know, workers in the colony that look fairly uniform, that look fairly similar to each other. But then there are a number of species in which the workers themselves have now started to split and turn into very different looking castes. And so over here, you have the large-headed soldier and the small-headed minor worker in the same colony. And for Darwin, this was a real problem because he couldn't figure out how it is that if natural selection is working on the level of the individual, determining reproductive success, how could you have in a single colony individuals with that are such dramatically different, that are such different in size to such a dramatic degree? And one of the things that we see is that this change that occurred in ant evolution, when the workers are looking so different from each other, resulted in incredible ecological success for those species. What you see here is a single colony. This is a leafcutter ant colony, and this is a single colony. You can see these are small humans over here. This is a single queens giving rise to this colony. This is in Texas. This is an atta colony. This is a picture taken by Lawrence Allen Smith. Here, a single ant colony center, you can see that it's wiped out all of the green all around it. And so this change to having multiple castes was quite dramatic. And it's not only the leaf cutters. Where's Anjali? Is Anjali here? Put up your hand, Anjali. She's the one who's in my lab working on these leaf cutter ants. She's traveling to Panama frequently in May, and she has her qualifying exam next week. You can wish her luck during the exam. But it's not only the army ants, the leaf cutter ants, but also the famous army ants, you know, with the soldiers with the massive jaws and the small workers. Or the marauder ants in the Philippines, in which you have these big, massive workers, and the smallest worker can almost stand on the head of the largest worker. Or the big-headed ants that we work on. This is the Phylogenetic, the large soldier and the small minor worker. Okay, and so attempts to try and explain this have largely occurred. This is a timeline from 1820 all the way to 2020. And these are, I'm not going to go into this in detail, but here's the Darwin's Origin of Species, where he raises the conundrum. And essentially through all these major events in the history of evolutionary thought and evolutionary science, this problem has gone unresolved. And what we started to realize, and these are collaborators of mine that work on these projects, this is Professor Daniel from the University of Arizona, and Christian Metzel. This is a very interesting story. He was, I met him on my first sabbatical in 2010. He was the guy who works at the front desk. And so for the first few months I was on sabbatical, I would just ask him about laundry and, you know, where do I do this and that. And one day we got into an interesting conversation. He asked me what I was doing, and I found out that he was a biologist. But he was just, he just wasn't interested in pursuing the field of biology. And so I started asking him, well, could you read these papers in German for me? And then he started to do so. And what we started to find, we started to unearth some really amazing things. And so together, on this right side line, completely unknown to these major events that happen in evolutionary science, this whole group of papers that were written in German, but were completely lost to science, probably because of World War II, because these were all pre-World War II, completely lost to science, revealed that Eco Evo Devo is almost as old as evolutionary science. Okay, so you've got, this is just what we looked at, Bloch-Mann in the 1880s. And actually had been going on, there's like a whole list of really active Eco Evo Devo researchers that had been going on in complete divorce from the mainstream of evolutionary biology. And what we've learned is that they've had a lot to teach us about Darwin's, the question that stumped Darwin. Okay? And also I'll go through a couple of our major discoveries and then how these workers have essentially, how they've enriched. And this is just a few of the studies, not even all of them. Okay, so what we work on is a remarkable genus of ants called Phyidole, or the big-headed ants. Again, like I said, they have these big-headed soldiers that do most of the defense. That's why they're called soldiers. And they help crack seeds. And the minor workers that do pretty much everything else. And they keep the minor workers at about 95% of the colony, and the soldiers at about 5%. And so our first big discovery we published in the journal Science, and you could see this fancy title, which we'll sort of decode. Ancestral developmental potential facilitates parallel evolution in ants. The day this was published on January 6, 2012. And this is, of course, my website. You can see that the number of hits on that single day just came out at just a spike. And then for a number of years after that, it was flat. And so I felt like I was defined by that single spike for many years. And the day that came out, it was a flurry of press that went into the news. The National Post, super soldier ants created McGill. BBC, ants turned into super soldiers. Nature, return of the super ants. PBS, Ehab Abouheif ants, Scientific American. And then it started getting even bad. McGill scientists create monster ants, Frankenstein ants created by scientists, giant-headed ants, technology haunt my nightmares, New York's horrific super soldier ants, we're doomed, welcome to a brave new world, and so on. It was just a flurry of news all over the world, and with massive headlines. So what's all that about? Did we really make crazy monster ants? Well, I'll tell you a little bit about the story, and we can decide. So there's a Phyidole. There's 1,000 species of Phyidole, all of which, most of them, have just this minor worker and soldier caste. However, there is 8 species of the 1,000 species, okay, which have at least 8 that have what's called a super soldier caste, okay, and they're only found somewhere in Arizona and northern Mexico. That's the only place you find these super soldiers. What do these super soldiers look like? They look like this. So here's, in these 8 species, so only 8 of the 1,000, you have a minor worker, here's a soldier, and here's these massive headed super soldiers, okay? And so that's one species. Here's what another species look like, comparing just the minor worker to the super soldier. Okay, and you could see why I might get an email begging me not to let the super soldiers out of my lab. But they're actually this tiny, so it's not really that worrisome. What do these super soldiers do? At least in one species, this is work from Nguyen that he published in a collaborator in Nguyen in 2012. So I showed you a picture of the army ants with the massive jaws. here's a small, this is a super soldier ant being attacked by army ants. And the army ants are nomadic raiders. Basically, when they sweep an area, they kind of go and they'll eat anything, including small vertebrates. They'll eat anything in their path. Okay? They kind of form and then they kind of come together. Okay. Here, you can see what they do is they use their big heads to block the nest entrance, and until all the army ants pass, and then they disband, essentially attack the army ants, and then form again their blockade. So this is what the super soldiers do. Now, one day I've been working on these Phyidoli, like one of the other 1,000 species without super soldiers, and I've been working on them in Long Island for at least 20 years now. And this is sort of the field we often collect. You can see it's very illustrious right next to the highway. And here's early students in the lab collecting them. This is how we dig them. One day, in a species that never has a super soldier, I'd never seen anything, we found this anomalously large soldier. Okay? And it took us quite a bit to recognize it. We were like, oh my God, that's a really big soldier. Is that what to do? What do you do with this big soldier? Is it really a big soldier? And so this discovery really got our, because we started connecting this big soldier now to the super soldiers that we knew about in Arizona and in New Mexico. Just to let you know, since that major discovery of this anomaly, this is what the site looks like now. So this, because this sparked the whole discovery, and now that's completely gone. There's just a massive mold on this one site where we found it. It's very heartbreaking. Okay, so basically here are these two species I showed you. One is called Phyidoli rhea. This is a soldier, that's a super soldier. And this is the tree, a Phyidoli tree. And there's another one on a separate occasion that has this super soldier. And this is where we found this enormously large soldier in this species that normally doesn't have the super soldier. What we knew about these things is basically this is what a queen looks like in the species with fully functional wings. That's a soldier, that's a super soldier. Now when a queen lays an egg, if it gets, this is usually a seasonal pulse. If it's part of the spring, it releases this hormone. And this hormone, if it detects it, it will turn into a queen. And it will develop, these two big things, these two big green things, which are the two forewings that you see here. If not, if it's just a normal part of the year, then it'll develop into a worker. Okay? And then this here, this is a nutritional switch. If the larvae gets enough nutrition, then this hormone is released and you become a soldier. If not, you become a minor worker. Okay? And that's how it works. Okay? It's kind of switch. So what we thought is what happens, since we saw this anomaly in the same species, what happens if we hit, this right before the soldier minor worker split? What if we hit it with a really high dose of hormone? Okay? And we did that, we were able to pop out something that looks exactly like the anomaly we saw, the super soldier-like anomaly we saw on Long Island, and that look like the super soldiers that you find in the wild. And this took us, by the way, to get this result took us about 8 years to find the right window where we could hit it, because you had to hit it with a very concentrated dose of hormone in a very narrow window. And when we did that, we were able actually to produce a super soldier in a species that doesn't have it. And we started doing that in other species, and we found that every time any species we touched without a super soldier, we could produce them by hitting them with these high doses, concentrated doses of hormone, and we could pop it out. What this means, essentially, and I'll just say it very quickly, is that in the ancestor, they had a super soldier, and even though the phenotype of the super soldier was lost on the outside, the genome inside the genes retained the potential to produce it for millions of years along this tree. Okay? So even though we might have, our ancestors had things and they lose them, we retain those potentials inside our genomes. And that with a really high dose of environmental stress or a hormone or an event, you can actually release those ancestral potentials and release the super soldiers. Okay? And so this was something, this was really the major point of the story, is that there's a lot hidden inside our genomes that we don't know about. Okay? And so one of the things, now coming back to the old literature, we had discovered the work of this guy Gutsch. This was, of course, after I had made this discovery, or it was actually meanwhile, so we'd made the discovery, we were writing the paper, and we realized that Gutsch had written, he was a German scientist, and this was an article written in 1939, and he had discovered that in these termites, most termites have soldiers, but there's one genus, so the ancestor has soldiers, because everything has soldiers, except for this one genus, it's called Anaplotermes. And in this soldierless genus, Anaplotermes, he found, like we found the anomalous individual soldier, he found a no soldier in this soldierless genus. Which means, again, it was the ancestor expressing its potential. And so Gutsch had made this discovery in 1939, and what we discovered, this is how we kind of got to it, that in the literature, this was in English in 1942 by an American scientist named Greg. He was discussing Gutsch's discovery. And so then I got Christian to translate Gutsch for me so we can go back and actually see the discovery that he made. It's really interesting because when he made the discovery, he was going on and how he found it, and then he was traveling across Argentina and how he kept it in his pocket and all kinds of crazy stuff. But if you see what Greg said, he says, it would mean that although soldiers do not appear in normal wild colonies and are supposed to have dropped out phylogenetically, their genes are in reality still retained by the species. This can be reinterpreted in favor of environmental control by assuming that while genes are admittedly present, some change in the physiological thresholds in the particular colonies in part enabled them to come to expression. The anomaly might also be interpreted as a result of phylogenetically suppressed genes, which cause the degeneration of certain characters, but which have not been entirely lost because of other vital influences in the termite colony. What that means is that they were already discussing it at that time. They had discovered these potentials, that they were trying to make sense of them. And what we were able to do essentially was to essentially show that those potentials can be converted into evolution. That evolution can use those raw potentials and convert them back into the evolution of species, which is something hadn't really been appreciated. And then we found another of Gutsch's paper in 1937, which was completely unknown to us, in which he connected for us that these potentials might be released by a variance in nutrition. And so he connected these switch points in hormones to actually nutrition, like especially this one, to a nutritional pulse. And so in this paper that you see here, he did crazy things. He did this thing where he said, therefore I commenced feeding liquid meat and solid sugar, i.e. frog and rabbit blood, meat juice, and raw egg white on one side, and hard breadcrumbs soaked in water sugar on the other side. After 4 weeks, soldiers developed only in cultures that had been fed meat chunks, whereas all larvae raised with liquid protein became workers. Later experiments carried out for half a year showed again that it was always a solid texture of the meat which is to be made responsible for the generation of soldiers. And he goes on and on. So he did all kinds of things: rabbit blood, frog blood, liquid protein, all kinds of stuff that you wouldn't necessarily get funding to do today. Okay? But he was able to connect the variance in solid protein, and he was messing with this. He was able to pop out a super soldier himself, but he just didn't know what to make of it. He couldn't figure out until we've now connected it just recently. That made us go back to Darwin now in a completely different book, and Darwin he didn't realize it, but he was actually almost came to the conclusion, the solution to his climax, but in another book in a completely different context. And so if we focus on this paragraph, scanned by Loren, thank you very much, from the Rare Books Library, it says here, the variation of animals and plants under domestication, Darwin writes, but on the doctrine of reversion as given in this chapter, the germ becomes a far more marvelous object. Besides the visible changes to which it is subjected, we must believe that it is crowded with invisible characters. So he's talking about inside ourselves, the invisible stuff you can't see, and to a long line of male and female ancestors separated by hundreds or even thousands of generations from the present time. And these characters, look how Darwin puts it, like those written on paper with invisible ink, all lie ready to be evolved under certain known or unknown conditions. So these proposals were all sitting around. People didn't know how to make sense of them, but now we're starting to appreciate what, this whole lost all these ideas that were lost, how we can make sense of them now. And now let me just do a thought experiment for you. What time is it? Oh my God, 6:15. How much time do I have left? That's great. Okay. I've lots to tell you, really exciting stuff to tell you. You gotta get to the vitamin T. I mean, this is crazy. Okay, so here, I want you to do a thought experiment with me. So now all this old stuff, what can we make of it? Now that we've shown that you can convert this invisible ink into evolution. We know now that climate change is rapidly changing. Okay? We know that we can't keep our domesticated crops. They're going, they're on the verge of extinction, and we're losing crops left and right because we've bred them so much to be productive that they're not that stress resistant. And so much of research now is going to try and inject genes from all over the place, from animals, other plants. They're GMOs. They're trying to inject genes into these crops, our domesticated crops, to make them more stress resistant. But what this perspective might tell us is that we know that the ancestors of all our domesticated crops were living in nature normally, and they were very stress resistant. Our point is that maybe some of that resistance is already there, hidden in the genome, but not expressed. And all we have to do, like invisible ink, is induce it, find a way to, actually produce these things, and then capture it in evolution. And we can bring back stress-resistant traits without any modification from, to the gene, unnatural modification to the genome. And Darwin, in his variation of domesticated crops, actually goes on about, how all these conditions of how you can release, like it's kind of crazy stuff. He found that when you breed things that are distantly related, it tends to release these ancestral kind of varieties. The other thing, just to say, what Gutsch did for us by showing it also exists in the termites, because when we discovered this in ants, we weren't sure if this was something that was unique to ants, or if it was a general character of all animals and plants, or all multicellular organisms. What we've now discovered, essentially, is the more we look, the more we find that these ancestral potentials are absolutely a universal property of multicellular organisms. And that's why this discovery is important. It's just that we were able to see it in the ants. What the ants allowed us to do, because it's kind of crazy, the Phyidoli, they're kind of like humans. They take care of their less fortunate. Okay? And so when there's an anomaly or something that doesn't look quite like the other ants, not a typical type of individual, they don't kill it unless it's infected. If it's infected, it's usually killed. If it's not infected, they actually feed it, they take care of it, and it just hangs around in the colony. And so from a biologist's perspective, we can actually see those variants. If they were just in solitary organisms, right, if there was like a frog or a rabbit, it might have got killed a long time ago, might not have been able to survive on its own. But because they take care of their unfortunate, we can actually see the action of what's being produced. These potentials, they give us an opportunity to see them. And so it was kind of like, how, very serendipitous that we were working on ants. It just happens that it makes this very obvious to see. And I think it's a very general phenomenon. Okay, let's move on to another key discovery with 15 minutes left. Let me see what I can do. Let's go. So this was another major discovery we made just last year. Okay, and this kind of this was in CBC, Montreal Biologists, Darwin's answer question, why do some ants become soldiers? And one of the things that really ticked us off from the old literature, because it wasn't really on our radar, is that another German biologist by the name of De Witts in 1878, what we discovered in his literature is he had actually responded to Darwin's climax. So in 1878, he was responding to Darwin. He wrote back, he was trying to talk about Darwin. He said, in his Origin of Species, Darwin speaks of the difficulty imposed onto his theory by the so-called neuters of eusocial insects. In particular, worker ants with a morphology differing greatly from reproductive animals, caused by the complete lack of wings. So all these dramatic differences in the colony again, he was trying to figure it out. This prompted me to investigate whether at least rudiments of these limbs, which are encountered so often in orthopterans, can be found. For a long time my search was in vain until I examined larvae and found imaginal discs at the side of the two back thoracic rings. What does that mean? It's just what DeWitt was trying to do is say, okay, you have these dramatically looking things. You have a miner worker, you have a soldier, you have a queen, and they all look so different from each other, right? And it's hard to connect them morphologically. How can we see, like, how can we draw the connections to understand their evolution? What DeWitt was saying is that, hey, let's look at development. Maybe in development we can find some characters that might connect them. And that's what we did. We said, well, the workers are all worker ants, like there's 15,000 species of ants, as I told you, and all of them are wingless, okay? But they all come from, all ants used to be, they come from winged wasps. So the workers, although they're wingless, what they do is he was looking for evidence of wings because that would connect the workers to the reproductives, and he found them. So this is a worker larvae, this is the head, this is the tail over here, and here what he's finding here is these little rudimentary wings. They'll never develop into the wings, but they just like pop up and then they disappear. So here you can see, these are going to become the legs, the 6 legs of any insect. And here you've got these little wing pads that will, in the queen and the males that develop wings, they'll become fully functional wings. But in the workers that are completely wingless, they appear for a very short time and then they disappear. Okay? And so that got us to think about something we had observed while we were looking at the super soldiers and discovering these potentials. And what we saw is that the minor workers, the small-headed guys, they didn't have it. These are the 3 leg discs that I just showed you. These will develop into the 3 pairs of legs. They had none. You couldn't see any. The soldier had 1 pair of this little rudimentary wing disc. The super soldier had these 2 massive ones. Again, they'll never develop into wings, but they have these little wing rudiments, these vestiges that just kind of pop up and for a long time, I just assumed that these vestiges were just, it was just when you hit with the hormone and activate the soldier or the super soldier program, I just assumed that these things were just consequences. They just, they just came out as a consequence of activating these programs. What we then discovered, and this was the big thing, is that if you, at this point, when the thing becomes a soldier, here's that single disc that I was talking to you about, that single to become a soldier, and it's a defining feature of the soldier. If you molecularly or physically ablate that disc, right? So either with molecular techniques, you go and you stop that disc from growing, or you go and you actually use the techniques that dentists use to burn the gums. We just put it on a larvae, and I can attest to you it's not that easy. Giovanna can tell us it's not that, and Alex can tell us it's not that easy, but you can actually try and ablate that disc. And when you do it, you can actually, this is what a normal, like under control conditions, that's what a disc looks like. We can actually reduce it, or we can eliminate it entirely. Okay? And when we did that, this was the crazy thing. This is a minor worker, this is a normal looking soldier. When you hit this little vestige of a wing, all of a sudden, you change the head and the body size of the whole ant. Okay, you can see here, this is the head dimension, and this is the whole body. This is just looking at the backside of an ant. And so this is what the ant would look like. So this is a normal minor worker, that's a normal soldier. You now change the head and the body. And so that kind of like, what the heck, this little vestige that's a wing, that would normally produce a wing, is now controlling the size of your head and the whole body? That's really kind of crazy that first of all, we think of vestiges as things that don't have any function. And now all of a sudden, it's regulating this, extraordinary morphology of the ant. And in fact, we can connect the soldiers and the minor workers. You can see here's the normal minor worker and the soldier. You can produce all kinds of intermediates that connect them just by messing with these little vestiges. Okay? Here's another view of the, of the vestige. Okay, so what does that tell us is that DeWitt in 1878 tipped us off that essentially there were these vestiges hanging around. He was the first to describe them. And he got us to rethink and look at what these vestiges might do. And actually, it's now pointing to, I mean, again, do this thought experiment with me. It turns out that all organisms, again, it's not one of these things that's strange about ant soldiers. All organisms, including humans, you have to think about your appendix, okay? You have to think, we have all kinds of features that pop up and disappear. So we actually have, during our development, we have a little tail that pops up and then disappears. We have webbing between our hands. We have gills that pop up and disappear. Okay? We have all kinds of features that are vestigial, that look like our ancestors, that pop up and disappear. Okay? And now, what we're saying is that we, in all of medicine, in all of biology, we've completely ignored them as things that might be completely functionless. Yeah, they tell us all of life is connected, but they're functionless. Now, this discovery is asking us to revisit all of that and think that actually these things might have a function in all of these different organisms. They appear, signal, and then they disappear. Okay? So that, I think, was really something. Okay, the last story I'll tell very quickly was this story. This is in collaboration with Moshe Sif's lab in McIntyre Med in the pharmacology department. And this has to do with the Carpenter ants. From these, basically, essentially, they, instead of just having just a miner worker and a soldier, they have miner worker, soldier, and everything in between. Okay? And so it's just a different kind of organization here. And this is the media coverage that came in. Canadian scientists double size of ants in experiment. This was my favorite. Canadian scientists bring about end of world with creation of giant ants. And then over here, why would you do this? Why would anyone do this? Okay, and so that was funny. And so so what did we actually do? So we did double the size of the ants. We managed to double the size of them. We managed to half the size of the ants. Okay, and how did we do this? It's a mechanism. We did this without changing the genes at all. Okay? It's something called the field of epigenetics. Okay? And with epigenetics, essentially, there are these chemical enzymes that chemically modify the DNA. Okay? And so they're called DNA methyltransferases. And what they do is when they chemically modify certain bases, certain code or bases on the DNA, they actually turn off the expression of those genes. Okay? And if you remove that chemical modification, you can turn it back on. Okay? And so it's not clear like how the chemical modification of these DNA and how you turn it back on. This is the field of epigenetics. And at that point, we weren't really sure if epigenetics could play any role. How do you actually do this? And so what we discovered, if you just look at these smallest miner workers versus these biggest soldiers, we look here at the degree of methylation. That's what this is. And you can see here that the miner workers have significantly more methylation than the big guys. And that makes sense because remember, when you chemically modify the DNA, you turn genes off. And so there's obviously some kind of, you turn genes off and you suppress to make a small guy, and you reduce the amount of repression and to make a big guy. Okay? And so when we did this, like I said, if you add methylation, so if you add methylation on, you chemically modify the genome of this ant, and you turn off gene expression, you can have the size of the ants. The green bars are the control, and the green is the experiment. You can see you shift the whole size. This is size on this axis, and the frequency. You can shift the whole size frequency down. Okay? If you then remove methylation, okay, and you let the genes get expressed, you can double the size of the ants. Again, the gray bars are the controls. You can shift the whole frequency that way, on that side. And most organisms have these chemical modifications. Now, the idea is that we know that nutrition has an effect on this, but we don't know how the nutrition interacts with this methylation machinery to make the environment interacts with this to make these chemical modifications and to have such dramatic effects on size. And if you think about it, I mean, if I was to array everybody in this room by size, we would form exactly this kind of size gradient. We would have like a frequency of small guys, you know, small people, and we would kind of go all the way to the tallest people, okay? I can tell you my grad student Junie is probably on this side over here. Okay, and so you can get this whole array, and we think that these, you know, epigenetic mechanisms might be at play together with genetic variation, you know, to determine all these inter-individual variations in populations. Now, what can Gutsch tell us? We went back to Gutsch in 1946, and it's crazy. Now, there was this whole, it turns out there's this whole episode between, you know, pre-World War I with Gutsch before, that, you know, was actually, he discovered something called vitamin T, okay? And he discovered it in termites, and it was eventually fed to children pre-World War I. And I'll show you exactly what happened. It's crazy. So he reported the discovery of this in 1946. The lineup of active agents has been extended by investigations that are now almost finished in bodies of some insects, especially termites, but also in several lower fungi, penicillium, hypomyces, and yeasts, have detected the presence of substances initially termed termitin or insectin, or penicin and taurolitolin. By now, all of these substances can be subsumed under the term vitamin T complex. We still don't know what it is today, but we know it's a certain substance. In all cases studied so far, from vertebrates all the way down to lower yeast, vitamin T stimulates assimilation. Cells stimulated by the agent gain the capability to mobilize existing reserves more quickly and to turn absorbed substances into their own living matter more efficiently. In other words, they can make fuller use of nutrients. Consequently, such cells grow quickly, proliferate more rapidly. This has, and of course, a different outcome in different species of animal or plant. Some lower organisms, such as yeast, fungi, and worms, multiply more quickly. In higher organisms, all processes of life are augmented, and it goes on and on. So he presented this discovery in 1946, and then he followed it up by experiments. Just to tell you, this is what he was reporting. So these are flies fed on a normal diet. These are flies fed on vitamin T. Okay? And you can see the effect in other insects. Okay, grasshoppers. Okay? These over here, these are this is cockroaches. You can see a normal diet, vitamin T diet. Okay? Just absolutely remarkable. Here's an experiment on white mice. So this is, for example, scarce food, no meat, without vitamin T, scarce food, no meat, with vitamin T. And you can see here the weight gain relative to the initial weight, and you can see there's quite a big difference. Normal food, little meat, no vitamin T, normal food, little meat, with vitamin T. Again, a big difference, and so on and so forth. Okay? And this is all in mice. So no matter what you feed this stuff to, it seems to have a major effect. And this was produced by industries, this vitamin T, T vitamin Gutsch, pharmaceutically, and it was fed to preemie babies because there was quite a bit of, you know, there was a problem with malnutrition after World War II, and so this is the use of vitamin T Gutsch in pediatrics. It was used extensively throughout Europe, in Germany, in Spain, in Italy, and so forth. And they say the abstract T vitamin Gutsch Pharmazil was given to 2 patients with celiac disease, 12 premature infants, and 28 children with dystrophy. Children improved but were receiving the same time the accepted treatments for their conditions, and so on and so forth. So this incredible thing just went kind of crazy. But now, I mean, what I think we're now at a point like, and then the whole thing died. After 1960, there was no mention of vitamin T and no mention of Gutsch, and the whole thing just died out. And we still don't know what the substance is, why it died out, what was the factors that led to it not going. And what our current hypothesis is now is that this vitamin T was actually the first probiotic that was ever discovered. And because probiotics are known, when people are malnutrition, probiotics have a huge effect on growth and weight gain. But when people are well nourished, the probiotics tend to not have a big effect on weight gain and growth. And so we think what Gutsch actually discovered, because it was in fungi, yeast, because if you can isolate this from yeast or different kinds of fungi, and he thinks that the termites were feeding on the fungi, because these are fungus-eating termites, they had this fungus in them in stored form. And so that's why we think it's some kind of probiotic. But that's our hypothesis. But and he's fed this to the ants, he's fed this to the Phyidoli too, and you just rapidly increase the percentage of soldiers. That's what happens when you give it to this. So we can actually now look back to see what this vitamin T was, whether it affects the epigenetics, whether it can double the size of ants, and we're kind of now on a new frontier here by looking back into the future. Okay, and just to finish now, so now that's just the whole, the whole idea of all these discoveries we've made and finding all of these, studies that were unknown essentially, or have been kind of relatively ignored from the mainstream of evolutionary theory. One wonders what lies in the dark, and where all of these forgotten ideas are, and where, the kind of work. I mean, they're hidden in Blacker Woods somewhere in German. I know Junie has a bunch of overdue library books that he's looking into now. So, so the ideas, they're there, hidden. But in this rapid pace of our modern, talk to any biologist in my department, Stephanie's here, can tell you, you can barely breathe. You can't keep up with all the modern literature. Things become old within 5 years. And what we do is we spin our wheels in current paradigms of scientific thought. So you're just going from one discovery to the next, and you're all working in our current paradigm. But there are tons of different paradigms that were hidden somewhere in different languages that raised really interesting observations and ideas that we need to connect with again, because maybe they didn't have the tools to answer them, but now we do have the tools to answer them. Okay, I think I've talked enough. Thank you very much for your